All right, lecture number 12 here. We're going to talk about what's going on in our capillaries as we exchange fluid and gases and nutrients with the tissues that they supply. All right, so capillaries, of course, are responsible for carrying blood to very, very specific local tissue places. And most of our tissues in the body are supplied with capillaries. We talked earlier in the chapter about the exceptions to that rule. Now, when we're thinking about gases and other solutes, they're going to tend to move by diffusion down their concentration gradients. That simply means that they move from areas where they're more concentrated towards where they're less concentrated. That's a concept that you learned early on in your anatomy and physiology courses. Okay, so how are different substances going to move between the capillaries and the surrounding tissue or interstitial fluids? All right, so if we're thinking about capillary structure here, we got a capillary passing through a tissue. Remember, a capillary is composed of very thin walls that are just one cell thick. So there's our capillary walls, and we got substances passing through the inside of the capillary here. Things that are lipids, lipid-soluble molecules, things that are hydrophobic, they can diffuse across cell membranes. Okay, because they don't mind the phospholipids in your cell membranes. They can get in there and mingle with those, so they diffuse directly across cell membranes. That's just like what you learned about in Biology 201. Lipid-soluble substances can directly diffuse across cell membranes. Water-soluble solutes, which that's most of the things that we study in most of the important molecules of life, biological molecules like water, they're hydrophilic or water soluble, so they don't freely move across cell membranes. They only get across when cells want to move them across. So those types of substances move through the clefts, the little spaces between cells and the walls of the capillaries, like we were talking about before, or the fenestrations. Remember, those are fenestrated capillaries and sinusoid capillaries actually have holes or openings in the walls, which make it easier for solutes to pass across. Larger molecules, things like proteins, for example, bigger organic molecules, the cells that make up the walls of the capillaries actually have to actively transport those from in here through the cell and out here into the exterior. So they will package them up inside little vesicles, transport those through the cell, and then open up on the other side of the wall and dump those substances out into the surrounding tissue fluids. All right, and here's just a review. Here's a picture of a fenestrated capillary like we were taking a look at earlier. So remember again, right in here, that's where you have those clefts, those little slits between the cells that can allow water-soluble solutes to move across. Now those slits are extremely small, so smaller types of solutes can get across. And by small, I mean things like amino acids, sugars, smaller chains of amino acids like some of your peptide hormones and so forth. They're going to be able to get through there, or they can move through the little pores there if you have fenestrated type capillaries. Bigger molecules, larger molecules like proteins, or bigger organic molecules, those are generally too large to move through those slits or those pores. That's why, as I mentioned, if you're supposed to move proteins, bigger molecules in here, out of a capillary and into surrounding fluids, they'll actually be taken up by the cells in the wall inside a little membrane-covered vesicle, transported across the cell, and then dumped out into the surrounding tissues. Okay, and this diagram from your textbook just shows you some of those processes that I was talking about on the last slide. I was just drawing them in for you. All right, fluid movement between capillaries. As you've learned about before, the fluids in your tissues are delivered by the blood. Fluid moves between compartments in the body. That's extremely critical to remember. All right, if we think about a capillary that is passing through a tissue, you know, out here you've got tissue cells in the tissue. You got fluid, obviously, in the capillary. How this works is, all right, and you got an arteriole over here and a venule over here, okay, because that's the way blood flows. And the way this needs to work, fluid and solutes need to move out of the capillary on this end, and they need to move back in over on this end. So on this end, we need the good stuff like oxygen gas. And hang on, my pen's not right again. I'm having lots of problems today with my little drawing tool here on my iPad. The iPad and the computer are not staying in contact with each other. There we go. On this end, we need to force the good stuff out. On this end, we need to take up the bad stuff along with some fluid as well. So how does that happen? How does that work so well? There are two forces that dictate how this happens. One of those is called hydrostatic pressure. And hydrostatic pressure is just fluid pressure. And it really is just referring to the blood pressure. The blood pressure, the force of the fluid against the wall of the capillary, there is some force associated with that, and that is going to help push fluids out through those slits between the cells and the capillary walls and through those little fenestrations if we're talking about fenestrated type capillaries. And then we have a different type of pressure, which is our osmotic pressure. And osmotic pressure is also going to, hang on. All right, so hydrostatic pressure involves blood pressure pushing fluids and solutes out. Then we have an opposing type of pressure that's called osmotic pressure, and that is due to solutes. Solutes attract water, and they can attract water across the walls of capillaries. Okay, and that results in what we call osmotic pressure. That's due to the movement of water by osmosis. And that's going to play a role as well. All right, so let's talk a little bit more about these hydrostatic pressures. And again, hydrostatic pressure involves the blood pressure in your capillaries, but then you also have interstitial fluid hydrostatic pressure, because you do have some fluid pressure within your tissues. Your tissues have boundaries around them. So the fluids in there are pressing up against those boundaries, and that results in interstitial fluid hydrostatic type pressure. All right, so your capillary hydrostatic pressure, which can be abbreviated as HPC, don't worry about that. That's the blood pressure in your capillaries. And what happens is, and if you think about a capillary passing through a tissue, it's going to be higher on the end near the arteriole. It's going to be lower on the end by the venule. So the blood pressure inside a capillary gradually drops from one end to the other. Now, you've already learned that because we've already talked about how blood pressure gradually drops from the arteries to the arterioles to the capillaries to the venules to the veins, and is that actually at its lowest as you return to the right atrium. But even within an individual capillary, that pressure is going to be 35, around 35, close to the arteriole, and it's going to be around 17 closer to the venule. So that means on this end over here, you're going to have more hydrostatic pressure pushing fluid out, and on this end, you're not going to have quite as much of that hydrostatic pressure pushing fluid out. Your tissue fluids, on the other hand, involves pressure that would push fluid back into a blood vessel. And that generally doesn't play a very significant role in fluid movement. And that's because we do have a system called the lymphatic system, which is a system of vessels that are not blood vessels. They're lymphatic vessels, and also includes things like your lymph nodes. And we'll talk about that in the next chapter and how that helps control your tissue fluid levels. All right, osmotic pressure is a sucking type pressure or a pulling type pressure that helps move. If we're thinking about a capillary here, we said you got hydrostatic pressure, your blood pressure forcing fluids out down at this end. And osmotic pressure is due to solutes. So if you guys remember, you learned about albumin proteins when we were studying blood. Those are large proteins, and you have a lot of those flowing through your bloodstream. Those do not get pushed out. They're too big to get pushed out of your capillaries and into surrounding tissues. But those proteins are solutes. They attract water. Water follows solutes. So that results in those albumin proteins suck water back in to the capillaries. And when you're over on this end, the venule end of a capillary, that osmotic pressure becomes so great that it tends to suck water back in out of the fluids and into the capillaries. The idea here is you need a balance. You don't want to push all of your fluids out into surrounding tissues, because then your tissues would just get bigger and bigger and bigger and bigger and bigger. You need to take up water back into the capillary so that you constantly have this back and forth exchange going on. All right, so this interaction between these hydrostatic pressure and osmotic pressures within the capillaries and the surrounding tissues results in something that's called the net filtration pressure. And filtration is referring to, you've got fluid and solutes passing through a capillary, and those things get filtered out or taken back up through the walls of the capillaries. That's a type of filtration that's taking place. And again, your net fluid flow is going to be out. More fluids coming out at this end than is coming back in. And down here at this end, it's the opposite. More fluid is coming back in than is moving back out. Now, the net result, as we're going to see, you really do push more fluid out at this end than you take up down at the other end, at the venule end. And that's where your lymphatic system comes in, because that would result in an excess of fluids leaving your capillaries and getting into surrounding tissues. So your lymphatic system kind of functions like a system of drainage ditches that help pick up that excess amount of fluid. All right, so let's take a look at this on a diagram. This is a good diagram for you to take a look at in your textbook here, figure 19.7. Bigger picture. All right, so here we have an arteriole that's bringing blood towards a capillary bed. It enters this met arteriole, if you guys remember that term from earlier, that comes off of this arteriole, and then that leads blood into a capillary bed. And at this end, and this is where we have our oxygenated blood, hydrostatic pressure inside the capillaries is high enough that it's pushing fluid and solutes out. As the blood keeps passing through here, your hydrostatic pressure is dropping and dropping and dropping as you move across the capillary bed. And when you get down to this side, when you're getting pretty close to the venule that's going to take the deoxygenated blood away, the osmotic pressure inside the blood, due to those proteins are attracting water back out of the tissue fluids, forces substances back into the capillaries at this end. So at the arteriolar end, fluid out; at the venule end, fluid back in. And at this end, Hydrostatic pressure is playing the biggest role in forcing fluid out. At this end, the osmotic pressure, the osmosis that takes place due to those albumin proteins in the capillaries, is sucking water or pulling water back in. Okay, and this diagram from your textbook tries to give you another type of visual on hydrostatic pressure. They've got a U-shaped container here with fluid on the inside and a boundary. So this would be like your capillary wall. And the piston here is supposed to represent like hydrostatic pressure. If you were pushing a piston down at that location, or like a syringe pump, that would push water across that boundary. So this is supposed to be like your capillary, and this is your tissue fluids over here. You push it across. And at the other end, down at the venule end, solutes attract water. So we have our boundary here. Those are our capillary walls. And those proteins become so concentrated down at the venule end that they're going to suck fluids back in down at the venule end. At the arteriolar end, you are pushing out things like glucose and amino acids and peptide hormones and other types of things that need to get to your tissues, oxygen gas. At the venule end, you're pulling back in wastes like carbon dioxide and other wastes that our cells generate, things like urea and uric acid. We'll learn more about those when we cover the kidneys towards the end of the semester. All right, this is a diagram which shows in more details what's going on. This is showing us at the arteriolar end of a capillary what's happening. So we have these opposing forces. Okay, so at the arteriolar end, you've got blood pressure in here, hydrostatic pressure, that is pushing fluid and solutes out of the capillary and into the surrounding tissue or interstitial fluids. That's about 35 millimeters of mercury, that pressure at the arteriolar end of a capillary. Now you do have albumin proteins in the blood all the time, so those do have osmotic pressure. Those albumin proteins are attracting water back in out of the tissues, and that is happening even at the arteriolar end. So you have fluid and solutes going out, you have fluid and solutes coming back in due to this osmotic pressure. But if you notice at this end, the hydrostatic pressure is higher, it's 35, than the osmotic pressure that's sucking fluid back in. That's only at 26. Okay, you do have a little bit of pressure from your interstitial fluids in here that's pressing up against the walls of the capillaries on this side, but it's generally considered to be about 0, and the lymphatic system plays a role in draining off any excess fluids and helping to maintain that as a negligible amount. Now you do have solutes. As you guys know, you have dissolved substances in your tissue fluids, but they're not as concentrated as they are in the blood. So you do have an osmotic pressure from the solutes in your tissue fluids that's sucking some water out of the capillaries as well, but it only contributes about 1 millimeter of mercury of osmotic pressure. So if you put all these things together, you got 35 pushing out, 1 millimeter of pressure being sucked out due to the solutes over here in your tissues. That gives you 36, and then you've got, that's opposing an osmotic pressure of about 26 that's trying to pull fluids back in at this end of the capillary. And that gives you an overall difference of the outward forces are 10 millimeters of mercury higher than the inward. So the net result is you tend to push fluid and solutes out of the capillaries at the arteriolar end. Now when you get towards the venule, your blood pressure has dropped across the capillary bed. That makes sense when you think about it. You've pushed a bunch of fluid out of the capillaries by this point, so lower volume leads to lower pressure. So down at this end, your hydrostatic pressure has dropped to 17 because you've pushed so much fluid out into the surrounding tissues. Your osmotic pressure, though, is still around 26. So that's sucking fluids back in. The hydrostatic pressure of your tissues is essentially considered to be 0. The osmotic pressure of your interstitial fluids is still around 1 down here at this end. But if you total all these things together down at the venule end, you have more pressure tending to move fluids and solutes back into the capillary as opposed to moving things out. You have a net movement in. So the result is down at this end, you're pulling in fluids. This is where things like the waste molecules, your carbon dioxide, your urea, your uric acid, they get picked up and moved back into the blood over here because the blood needs to transport those things away so you can excrete them out of your body. All right, so that was just some more information about, well, how does this even work? How do the capillaries perfuse the tissues and drop off the good stuff and pick up the bad stuff? And that's how that works. It's due to this competing hydrostatic pressure versus osmotic pressure between the blood and the surrounding tissue fluids. The final lecture for Chapter 19, we're going to talk a little bit more about a homeostatic imbalance, circulatory shock that can occur with extremely low blood pressure, and that will finish up Chapter 19 for us.